Hi, hello my dear students. Welcome to our channel Rajesh Biology, and I am Rajesh MSc Biology. Before starting the session, let me know if our session is started or not, and if my voice is clear or not first. Okay. Yes. So still session is not yet started, I think. If it is started for you, try to mention in comment box as it is started. Okay, it is started now. So if my voice is clear, let me know if there is any disturbances. Okay. Right. So without any late, we are going to start the session. First of all, a interesting fact about the Great Wall of China. You may already know it is the one of the wonders of the world. Okay, and this is built for 6,400 kilometers length, and it is the longest man-made wall in the universe, in the world. Okay, and why it is built? That is the question. ఇది ఎందుకు బిల్డ్ చేశారు? ఇంత పెద్ద వాల్ని బిల్డ్ చేయాల్సిన అవసరం చైనాకు వచ్చిన అవసరం ఏంటి అంటే వీళ్ళకి నార్థన్ ఏరియాస్లో ఉండే కొంతమంది కొన్ని దేశాల నుంచి రక్షణ కోసం నిర్మించిన ఒక పెద్ద వాల్. ఇది గ్రేట్ వాల్ ఆఫ్ చైనా ఈజ్ ఓన్లీ ఫర్ ద ప్రొటెక్షన్ ఫ్రమ్ ద ఇన్వాడర్స్ ఫ్రమ్ ద నార్థన్ కంట్రీస్, ఫ్రమ్ ద నార్థన్ ఏరియాస్. ఓకే, దిస్ ఈస్ ద రీజన్. Now come to the first question in our session, that is the final proof for the DNA as the genetic material came from the experiments of what? Good evening, Saujanya. Very good evening. First of all, have you completed your supper or not? Supper means dinner. Everybody. First of all, have you completed your supper? Yeah, Evergreen says is A. Evergreen says A. Hershey and Chase, Avery, MacLeod and McCarty, and Hargobind Khorana, Griffith. Who are all these persons? If Hershey and Chase are the right answer for this question. Yes, what you said is right, Evergreen. The right answer is Hershey and Chase. I don't know what is the reason. My notepad is not working properly. Hershey and Chase is the right answer for this question. The right answer is A. Hershey and Chase. They conduct the experiment on the bacteriophage. Okay, bacteriophage means what? Bacteriophage means virus. Bacteriophage means the virus which infects the bacteria, that is called bacteriophage. And only two structures are present in bacteriophage. One is a protein coat, another one is DNA material. So depending upon this simple principle, Hershey and Chase finalize and they give the final proof as DNA is the genetic material. Right, Muhammad Farkan, perfect, and Saujanya, very good. That is A. A is the right answer. Come to the second question. Come to the second question. If there are 999 bases in a RNA that code for a protein with 333 amino acids, you already know what is codon. Codon means what? A series of 3 amino acids is called codon, and that codon responsible for one amino acid. If there is 999, 333 amino acids, perfect. But the question is, and the base at the position 901 is deleted. 901 is deleted such that the length of the RNA becomes 998. How many codons will be altered? How many codons will be altered? Try to mention the question number now. Please try to mention the question number. Here, 999. Codon means what? A set of 3 nitrogen bases is called codon, and it is responsible for it. Codon codes for one single amino acid. So 999 codons responsible for the formation of 333 amino acids. But now question is, at the 901 position, one amino acid is deleted. One nitrogen base is deleted. If one base is deleted, how many are there? So up to here 999, up to 900 everything is correct, perfect. 900 means 300 amino acids are never change. From the 901, that means the remaining 33 will altered. So for the second question, the right answer is B, 33. Let me know if it is any question, if there is any question with this problem. Okay, I'll definitely explain this. First of all, you have to understand that codon is a triplet. Codon is always triplet. Maybe it is AUG or something, GUG, AUU or GUU, like that anything. And that is responsible for the formation of one amino acid. Okay. And the question is 999 nitrogen bases are there. Okay. If we divide by 3, it is 333 amino acids will be formed from the 999. If 901, the position of 900 first position is deleted, the remaining amino acids will be altered. What are the remaining? There is no problem with the 900. That means 300 are perfect. But the remaining amino acids are altered because of the deletion of 901 amino acid. So the remaining are 33 because 333 minus 300 is equal to 33. The right answer for this question is D. Yes, actually participate. Try to mention the comment as it is okay, it is understood, or I know, but I can't explain the reason. Like that, you can mention. Okay, try to say the at least wrong answer. The association of histone H1 with the nucleosome indicates that, very easy question. The association of histone H1 with a nucleosome indicates what? What is nucleosome? Nucleosome means histone octamer. Histone octamer, and it is bind with one H1 protein, and around this DNA is present like this. It is the interlinked DNA. Yes, Saujanya, please tell me. Yes, thank you, Saujanya. Okay, Sarah. Right. So for the third question, can you say the answer for the third question? As a biology students, as a NEET aspirants, we should develop interest in this type of chapters like molecular basis of inheritance, principles of inheritance, which are more complex, they are more interesting. Yes, Evergreen says the DNA is condensed into the chromatin fiber. Perfect. The association of histone H1 with a nucleosome means what? It is condensed as a chromatin fiber. We can observe this in a chromatin fiber only. Okay. DNA replication lo nucleosome undadu. And DNA double helical is exposed. Expose ayindi ante there is no nucleosome structure. DNA simple ga undani artham. Transcription time lo kuda DNA simple ga untundi. There is no condensed form. At the time of condensation only there is a nucleosome. దీని క్లియర్ స్ట్రక్చర్ న్యూక్లియోజోమ్ అంటే ఏంటి, క్రోమోజోమ్ అంటే ఏంటి, క్రోమాటిన్ అంటే ఏంటి, సొలనాయిడ్ అంటే ఏంటి, డిఎన్ఏ అంటే ఏంటి, ఇవన్నీ మనకి రాజేష్ బయాలజీ ఛానల్లో జెనెటిక్స్ అనే ప్లేలిస్ట్లో ఉంటుంది. ఓకే, మీరు దాంట్లో అబ్జర్వ్ చేయొచ్చు. నెక్స్ట్ 4th క్వశ్చన్. ఏ మాలిక్యూల్ దట్ కెన్ యాక్ట్ యాజ్ జెనెటిక్ మెటీరియల్ మస్ట్ ఫుల్ఫిల్ ద ట్రైట్స్ గివెన్ బిలో ఎక్సెప్ట్. Genetic material fulfill the traits given below except one. Except what? Yes. See, it should be unstable structurally and chemically. Perfect. Very good, Evergreen. It should be stable, not unstable. A genetic material should be stable. That's why when compared with RNA, DNA is more stable. That's why DNA is the good genetic material because it's stability. So stability is the nature of the genetic material. But it says as it should be unstable. So this is wrong. And this is the right answer for this question. It should be able to express itself in the form of Mendelian characters, right? It should be able to generate its replica. It is also right. It should provide the scope for slow changes that are required for evolution. That is also right. Okay, yes, Bhargav, very good. The right answer is the C. It should be stable, not unstable. So the right answer is C. Next question, 5th question. DNA dependent RNA polymerase catalyzes transcription on one strand of the DNA, which is called. What is the strand which gives the information for the RNA? There is double-stranded DNA is there. Okay. And RNA is prepared from one of the strand. This is RNA. This is prepared from one of the strand, A and B. For example, gives the information. B transcripts the information. What is this strand called? Evergreen. Template strand. Perfect, Bhargav and Evergreen. From now onwards, try to mention the question number, please. Or otherwise, I may thought as your answer is for the previous question. Please mention the question number. Template strand is the right answer. Very good. Next, which one of the following is the starter codon? Starter codon. Every year start with August. English year start with August. No, English year start with January. Telugu year start with April. Biological year start with August. Like this. Yes, very good. Bhargav, 6th one is AUG, correct. AUG is the starter codon. And it is form amino acid also, methionine. Okay, that is also. Yes, Evergreen, 6th one C. Come to the 7th question. Which of the following is required as inducer? Inducer. Which of the following is required as inducer? Which of the following is inducer? Okay, 1 minute. I'm going to share this in our Telegram group. Right. Seventh one says A and Bargo says D. Inducer is lactose. Perfect. Evergreen. Lac operon. Actually, lac means lactose. Lac operon, lac means lactose. Lactose is the inducer. Very, very important. Okay. In the absence of lactose, the repressor bind with the operon or operator. So lactose is the inducer. Next question, eighth question. A complex of ribosomes attached to a single strand of RNA is known as. A complex of ribosomes attached to a single stranded RNA is known as. It is very, very easy question. Important question. Complex of ribosomes. It is a repeated question. So many times it is repeated. Complex of ribosomes attached to a single strand RNA. It is known as. Eighth one. Very good, Bhargav. It is C. Polysome. Remember this point because it is a repeated question. Same question will repeat in a different manner in the next slides. Which one of the following is not applicable to RNA? Yes. 8th C, correct. Evergreen. Evergreen name I forget every time. Harika. Which one of the following is not applicable to RNA? Heterocyclic nitrogenous bases. No, it is applicable because RNA also have the purines and pyrimidines. Ninth question. I want answer for the ninth question. Chargaff's rule. What is Chargaff's rule? Who is Chargaff? Is he our neighbor or our school principal? Or he is the scientist who give the information or structure of, who describes the structure of DNA? Chargaff is the structure of, he is the scientist who describes the structure of bDNA. That rules are applicable only for the bDNA, not for the RNA. So, this is the right answer, Chargaff's rule. Chargaff's rule is what? That is 34 angstroms length, one helix, and between two adjacent nitrogen bases, 3.4 angstroms, and adenine bind with thymine with 2 hydrogen bonds, guanine bind with cytosine with 3 hydrogen bonds. This is Chargaff rule, and it is only responsible or applicable for DNA. 10th question. Which one of the following hydrolysis internal phosphodiester? I want to mention, it is worth to stress on this word. Internal phosphodiester bonds. Hydrolysis of internal phosphodiester bonds in a polynucleotide chain, whether it is RNA or DNA. Which enzymes or which one of the following hydrolysis internal phosphodiester bonds? Participants are more, but why only 2 are actively participate? Evergreen and Bargo. Which one of the following? Yes, Bargo. 10D. Exonuclease. Nuclease is right because nuclease is the enzyme which hydrolyzes the phosphodiester bonds in polynucleotide chain. Nucleases are of two types, exonuclease and endonuclease. Whatever the type is, nuclease is responsible for the hydrolysis of phosphodiester bonds. Means what? Adenine and thymine, these bonds are breakdown and these bonds are breakdown. Okay, nucleotide convert into nucleoside, nucleoside convert into the sugar and like that. So Here, the most important thing is internal. It is endonuclease. Yes, perfect. Balbiani rings are the sites of. Maybe it is new to you. Try to answer this question, or otherwise I'll reveal this. Balbiani rings are sites of. There is a chromatin structure of RNA like this. This is actually Balbiani ring. Most of the insects RNA synthesis, okay, and protein synthesis areas, there is a site, a ring-like puff-like structure. There is a puff-like structure, and this puff is called Balbiani ring, where the RNA and protein synthesis takes place. Of course, very good, Bhargav. That is B. Come to the 12th question. Maybe that is not present in our NCERT textbooks or IP textbooks, I don't know, but it is present in reference books. Okay, no problem with that question. If you don't know about that, just forget about, don't bother with the type of typical questions. Come to this question, which is very easy. Which of the following statements is not true of two genes that show 50% recombination frequency? 50% recombination frequency. Bhargav and Evergreen, have you completed your dinner or not? And my dear subscribers, sorry for the late live sessions. టైం కుదరడం లేదు, బట్ కుదిరిన ప్రతిసారి ఫస్ట్ ఆలోచించేది మీ గురించే. ఐ వాంట్ టు కంప్లీట్ ఎంటైర్ సెకండ్ ఇయర్ చాప్టర్స్ ఇన్ ఏ సిరీస్, ఒక ఆర్డర్లో కంప్లీట్ చేద్దానికి ట్రై చేస్తున్నాను. సో నెక్స్ట్ చాప్టర్ ఎవల్యూషన్. దెర్ ఇస్ నో ప్రాబ్లమ్ టు సే ఎస్, వెరీ గుడ్. We're going 12th D. So, which of the following? Everyone says SD. The genes are tightly linked. Very good. When the genes are tightly linked on the same chromosome, they cannot form recombinations, because how much they are close, how much they are close, based on that, if they are more close, the recombinations are more or less. If they are far from one another, the recombination number is more. So the genes which are tightly linked, they cannot form 50% recombinations. It is the theory of recombination. Now come to this question. Which enzyme will be produced in a cell if there is a nonsense mutation in the lac Y gene? Very interesting question it is. Okay, it is beta. Okay, don't confuse with the box. It is not a box, it is beta galactose. Beta galactosidase. Okay, it is not box. Now question. Which enzyme will be produced in a cell if there is a nonsense mutation in the lac Y gene? You know, there is 4 genes are there: I gene, Y, Z and A. Actually, this is the order: Z, Y, A, I, Z, Y, A. And he asked about if there is a mutation in the Y gene, okay, which enzyme will be produced? That is C. Very good, Evergreen. That is beta galactosidase. Beta galactosidase is produced if there is any mutation in the lac Y gene. How? Actually, beta galactosidase is not produced by the Y gene. It is produced by which gene? Z. It is produced by Z gene. Then how Y gene get mutated? Only galactosidase is present, because after I, first lac Z will be gives this information. That means galactosidase is produced. Later Y, it is already mutated, so it never produce any enzyme. Because of the mutated gene, it will close, the operand concept will be closed. So A is also not yet produced. Permeases or stelase, they are not produced. Beta galactosidase is produced because before the Y expression, Z is expressed out. So only beta galactosidase is present. Okay, lactose permease is produced by the Y, but because of the mutation, it is not produced. Transacetylase, it is also produced by the A, but it is also not produced because of the mutation. Operand concept is closed. Okay, so 13th C. Come to the 14th question. I hope you understand what is going on in the lac operand concept. If there is a mutation in Y gene, if there is a mutation in Y gene, it will be stopped. Alright. Removal of removal of introns and joining of exons in a defined order during the transcription is called. It is a direct question from the NCERT lines. I forgot the page number, but it is very, very important, repetitive and direct question from the NCERT lines. Looping, inducing, slicing, splicing. Yes, very good, Bhargav. Splicing it is. Removal of introns and joining of exons in defined order during the transcription is called splicing. Bhargav, you are going to crack the NEET examination for the next NEET. Okay, because the questions which we answer, the questions which are from the NCERT textbooks, if we can answer that questions, we are done with more than 70%. 70% కన్నా ఎక్కువ మనం బాగా ప్రిపేర్ అవుతున్నామని అర్థం. డైరెక్ట్ క్వశ్చన్స్ మనం ఆన్సర్ చేస్తున్నామంటే ఎన్సీఆర్టీ నుంచి. Okay, the remaining 30% based on your logical skills, because out of the NCERT questions, impossible, mostly impossible. Depth వెళ్ళొచ్చు కానీ కొంచెం అటు ఇటుగా. సో 70% మీరు బిట్స్ ఆన్సర్ చేయాలంటే ఎన్సీఆర్టీని తరువుగా రిపిటేటివ్గా చదవడానికి ట్రై చేయండి. కమ్ టు ద 15th question. Try to answer the 15th question. Removal of RNA polymerase 3 from nucleoplasm will affect the synthesis of. Will affect the synthesis of what? Yes, Bhargav, that is tRNA. Polymerase 3 is responsible for the formation of some tRNA and proteins like that. So tRNA is affect. Yes, Saujanya, please welcome back. Okay, it is not HRNA, it is tRNA. Come to the 16th question. But well tried, Saujanya. Try to answer this question. What are the structures called that give an appearance as beads on string? Beads on string in the chromosomes when viewed under electron microscope. Very important, interesting, easy question it is. Beads like this, present on a string like this. Which structures are these to give this type of appearance? What are these beads? Yes, Bhargav. Manju Bhargav says C. Evergreen says C. Nucleosomes, everybody knows as it is C. Right? Nucleosomes, all these are nucleosomes. Histone octamers. The unequivocal proof of DNA as the genetic material came from the studies on the unequivocal. Unequivocal means దీని మీనింగ్ ఏంటంటే ఇంకా తిరుగులేనిది అని, ఇంకా తిరుగులేని ప్రూఫ్. Unequivocal proof of DNA as the genetic material from the studies on. We already discussed. I think this is the first question from this quiz. ఈ క్విజ్లో మొట్టమొదటి క్వశ్చన్ ఇదే, దీని రిలేటెడ్. Who gives the final proof? That final proof is unequivocal proof. And they conduct the examination, they conduct the experiments on bacterial virus. Very good. Evergreen. Sorry, Bhargav says, right answer, bacterial virus, not the bacterium. It is a bacteriophage. Bacteriophage means virus. We already discussed. Bacteriophage means bacterial virus. Okay, right. Now come to the next question, 18th question. Which one of the following does not follow the central dogma? Actually, of course, maybe 1 year back, I think, I received a meme. I observed a meme in the Instagram. In that meme, one boy and one girl chat with one another, and girl asked to that boy, baby, please tell me the three magical words. And that girl expects I love you, but the boy who is biology student gives the information, gives the reply as replication, transcription and translation, because for our biology students the magical words are this transcription and translations. Okay, really excellent phenomena. If we have interest in the genetics field, we just love the genetics more than anything. So for this question, Manju and Saujanya says HIV. Perfect. Because HIV is reverse to the central dogma. It is not from DNA. It is from RNA to DNA. It prepares a DNA by using RNA. Okay. Reverse transcriptase. Come to the 19th question. Whose experiments cracked the DNA and discovered unequivocally that a genetic code is a triplet? Whose experiments cracked the DNA and discovered unequivocally the genetic code is a triplet? Who is that scientist? Manju Bhargav says as Hershey and Chase. Unequivocally and చూసిన వెంటనే మనం Hershey and Chase అనుకోవద్దు. హర్షీ అండ్ చేస్కి ట్రిప్లెట్ కోడానికి అసలు సంబంధం లేదన్నది నా ఉద్దేశం. జెనెటిక్ కోడ్ ఇస్ ఏ ట్రిప్లెట్ అని ఎవరు చెప్పారు? తిరుగులేని విధంగా జెనెటిక్ కోడ్ అనేది ఒక ట్రిప్లెట్ అని డిస్కవర్ చేసిన సైంటిస్ట్ ఎవరు? దే ఆర్ నన్ అదర్ దెన్ నిరెన్బర్గ్ అండ్ మెథాయ్. నిరెన్బర్గ్ అండ్ మెథాయ్. Yes, okay, no problem. 20th question. The one aspect, yeah, Manju Bhargav, that is D. Nirenberg and Mathaei. And okay, Saujanya, that is D. Come to the 20th question. The one aspect which is not a silent feature of genetic code is its being. The one aspect which is not a silent feature of the genetic code is its being. Genetic code feature కానిదేంటి? Bhargav says the specific. No, genetic code is specific, highly specific genetic code. Specific means what? If there is a genetic code, AUG is there, it is responsible for the methionine. It is the specific for the starting codon, like that. Specific it is. It is universal. If AUG is the starting codon in this organism, that is the same is responsible for the same methionine and starting codon for the other organism also. It is universal. It is same in my body and your body, in all the persons, humans and organisms body. It is universal. And it is, yes, it is universal. Which is not, you have to focus on this, which is not a silent feature of genetic code. It is universal, it is specific. So these two are not right answers for this question. You have to choose any one from A and B. The right answer is, for the 20th question, ambiguous. Genetic code is not ambiguous. Genetic code is not ambiguous. Sorry, Evergreen. Okay, Manju Bhargav is right. Maybe it is a flip coin. If it is head, degenerate. If it is tail, ambiguous. Like that maybe. But okay, let me explain what is degenerate and what is ambiguous. Degenerate means what? Degenerate means, for example, just for example only I am giving. Okay, GUG is responsible for one type of valine. For example, valine. GUG is responsible for valine. Valine is also produced by the AGG. Valine is also produced by the GAG. This is degenerate. This character is called what? Degenerate. Ambiguous means what? It is actually non-ambiguous. It is unambiguous. Ambiguous means what? Ambiguous means GAG produce valine, GAG produce serine, GAG produce lysine also. If it is like this, ఇలా ఉంటే దాన్ని ambiguous అంటాం, but it is not correct. One triplet, one genetic code responsible only for one amino acid, that's it. So it is unambiguous. Genetic code is unambiguous, but ఇక్కడ ambiguous అని ఇచ్చారు కాబట్టి wrong answer, ambiguous. అర్థమైంది కదా? డిఫరెంట్ GGA, GGU, సారీ GGU ఉండదేమో కదా, GGA, GGT, ఓకే, ఆర్ GGC, TAT, ఈ మూడు ఒక ప్రోటీన్కి ఇవ్వచ్చు. ఈ మూడు ఏది వచ్చినా సరే ఒక ఎలనైన్ ఆర్ ఏదైనా ప్రిపేర్ అవ్వచ్చు, డీజనరేట్. కానీ TAT అనే దానికి డిఫరెంట్ వాటిని క్రియేట్ చేసే కెపాసిటీ లేదు. ఓన్లీ ఒకదానికి మాత్రమే అది కట్టుబడి ఉంటుంది అనమాట. సో ఇట్ ఈస్ నాన్ ambiguous, బట్ ambiguous ఇచ్చారు కాబట్టి దిస్ ఈస్ ద రాంగ్ స్టేట్మెంట్. 21st question. So yes, thank you Avery. Select the 2 correct statements out of the 4 statements given below about the lac operand concept. Very interesting, very easy concept. Glucose or galactose may bind with the repressor and inactive it. Bind with the repressor and inactivate it. Glucose and galactose. Correct? No, it is lactose actually, not glucose or galactose. In the absence of lactose, the repressor binds with the operator region. ఇది మనం ఇందాకే చెప్పుకున్నాం. If there is no lactose, repressor bind with the operator region. So second one is right. So according to this, except B, maybe it is A, C or D. 21st says C. Perfect, Bhargav. C is right answer. 4th one is, it was elucidated by the Francois Jacob and Jacques Monod. 4th one is also right. 2 and 4 is present in the C, option C. Okay, the judging codes for permease, it is wrong. That is beta galactosidase, right? 22nd, in the DNA molecule, the proportion of the adenine in relation to thymine varies with the organism. Yes, Evergreen. DNA molecule adenine relation with thymine is vary from organism to organism? No. In every organism, adenine bind with thymine, that too with double bonds, 2 hydrogen bonds. So first option is wrong. There are 2 strands which are anti-parallel, one in 5 and to 3 n direction and other in 3 and to 5 n direction. So this is right. Let me read the another statement also. The total amount of purine nucleotides and pyrimidine nucleotides is not always equal. This is also wrong. The purine nucleotides are always equal to the pyrimidines. No, Evergreen. 22nd, C is wrong. DNA molecule have equal number of purines and pyrimidines. There are 2 strands which run parallel in the 5 to 3 direction. This is also wrong. If one is 5 to 3, another one is 3 to 5 end. So the right answer is B. Perfect. Saujanya, very good. Great. Come to the 23rd question. Which one of the following statements about the particular entity is true? Particular entity is true. Centromere is found in all animals, correct? All animal cells centromere. Is this correct? Centromere is found in all animal cells which produces ester during cell division. Okay. What is ester, you know? And next, the gene for producing insulin is present in every body cell. Yes. Bargo says A is right. Okay. Nucleosome is formed of nucleotides. Nucleosome found in nucleotides. DNA consists of core of 8 histones. So, no Bargo, A is wrong option. Centromere is not there. It is not centromere, it is centriole. It is what? It is centriole. Centriole is present and the centriole is responsible for what? 1 minute. Centriole produces this type of asters. During the cell division, asters are there. Okay, asters from there, spindle fibers are there. Spindle fibers attached with the centromere, centrosome, like this. So these are called asters, which are formed by the centrioles. These are what? Centrioles during cell division. So this is wrong. Next, the gene for producing insulin is present in every body cell. It is right. Every body cell have. Yes, Pujita. Welcome, Pujita. Good evening. You are at 23rd question. Try to answer the remaining 7 questions. Every cell have insulin producing gene. Perfect, because every cell has a nucleus, DNA, 23 pairs of chromosomes are there. So insulin producing gene is present in every cell. This is right. Nucleosome formed by nucleotides wrong. Nucleosome formed by the histone proteins. DNA consists of core of 8 histones. This is wrong. DNA consists of nucleotides. Okay. So the right answer for this question is which one? Option B. 24th. Pujita, have you completed our supper? You know one thing, the food what we intake at night time, that is called supper, not dinner. Dinner అని మనం ప్రొనౌన్స్ చేయదు ఈసారి నుంచి, అది సప్పర్ అంటాం. మరి డిన్నర్ అంటే ఏంటి? ఇఫ్ ఎనీ పర్సన్ ఇన్వైటెడ్స్ టు దేర్ హోమ్ టు టేక్ మీల్స్, దట్ ఈస్ డిన్నర్. మనం వేరే వాళ్ళ ఇంట్లో చేస్తే డిన్నర్ అంటాం, మన ఇంట్లో మనమే తింటే దాన్ని సప్పర్ అంటాం. ఓకే. ఎస్. Saujanya B. Pujita, completed your supper. Which one of the following pairs of nitrogenous bases of nucleic acids is wrongly matched? 24th. For 24th. Guanine adenine, purines perfect, AG or purines. TC, thymine and cytosine and uracil, these are pyrimidines. So the right answer for this question is B. Okay, very good. Adenine, thymine, these are not purines. Adenine is pyrimidine, adenine is purine, thymine is pyrimidine. Perfect. Who is that? Evergreen. Very good. Vijay Yadav, why don't you mention the question number, Yadav? Vijay Yadav. From where, Vijay Yadav? You are from which state or which district? Polysome is formed by, I'm already mentioned, I think, Evergreen. ఇంతకు ముందు మీరు ఆన్సర్ చెప్పారు, భార్గవ్ అండ్ ఎవర్గ్రీన్ ఆన్సర్స్ చెప్తున్నప్పుడు నేను చెప్పాను. It is very important repetitive question. It may asked several times in different ways. Polysome is formed by a ribosome with several subunits. Ribosomes attached to each other in a linear arrangement. Manju Bhargav, perfect. Several ribosomes attached to a single mRNA, that is polysome. Yes. NEET రెగ్యులర్గా ఉంటుందా సార్? నీట్ రెగ్యులర్గా ఉంటుందా అంటే ఉంటుంది. ఎక్కడ, మన లైవ్ సెషన్స్లోనా లేకపోతే గవర్నమెంట్ రెగ్యులర్గా నీట్ని ఎగ్జామ్ కండక్ట్ చేస్తుందని అడుగుతున్నావా? క్వశ్చన్ కంప్లీట్ చేయి పూజిత. మన సెషన్స్లో నీట్ రెగ్యులర్గా ఉండడానికి నేను ట్రై చేస్తున్నాను, బట్ నాకు కుదరడం లేదు. ఎందుకంటే కాలేజ్ టైమింగ్స్కి లైవ్ సెషన్స్ పెట్టడానికి వేరే ఉండే ట్యూషన్స్కి వీటికి సెట్ అవ్వడం లేదనమాట నాకు. బట్ ఐ విల్ కంప్లీట్ ఆల్ ద చాప్టర్స్ బిఫోర్ ద నీట్ ఎగ్జామినేషన్. ఐ విల్ ట్రై టు కంప్లీట్ ఎట్ ఎనీ కాస్ట్ అండర్ ఎనీ సర్కమ్స్టాన్సెస్. ఇంకా మనకి కొన్ని చాప్టర్స్ ఉన్నాయి. దీని తర్వాత చాప్టర్ అయిపోతే బాటనీలోకి ఎంటర్ అవుతాం మనం. సో మ్యాక్సిమం కంప్లీట్ అయిపోతాయి. ఒక 32 ఉంటే ఇది 28 కాబట్టి ఇంకొక 5 or 6 ఉంటాయి. యూ విల్ కంప్లీట్ ఇట్ లైవ్. రెగ్యులర్గా ట్రై చేస్తాను. 26th ఆన్సర్ చేయండి. భార్గవ్ సేస్ విచ్ వన్ ఆఫ్ ద ఫాలోయింగ్ పేర్స్ ఆఫ్ కోడాన్ ఈస్ కరెక్ట్లీ మ్యాచ్డ్ with their function or the signal for the particular amino acid. Okay, very good Bhargav. Saujanya and Pujita says C. GUU, GCU, alanine. No. Evergreen and Bhargav says they are right. UAG, UGA, and another codon is also there. Can you say that? These three are stop codons: UAG, UGA, UAA. These three are stop codons. So D is the right answer for this question. Yes. 27th question. Pujita, ఇప్పుడు నువ్వు ఫస్ట్ ఇయర్ సెకండ్ ఇయర్ అమ్మా? Twenty-seventh question. One turn of the helix in a B-form DNA. You know, different types of DNA molecules are there. Nearly more than 20 are there. And we give the alphabets to the DNAs depending upon their particular structure. Okay. But in intermediate, we are going to study about the B-form DNA, which is more important and more abundant. Okay. So for this B-form DNA, Chargaff gives some rules. According to the Chargaff rules, B-form DNA, one turn of the helix is approximately, what is the length of the one turn of the helix? It is actually called a pitch. Okay, this is helix turn. To complete one turn, what is the length of this one turn? Approximately. First year కదా పూజిత, నో ప్రాబ్లం. నువ్వు సెకండ్ ఇయర్కి వచ్చేటప్పటికి యూ విల్ బి రెడీ ఫర్ ద నీట్ ఎగ్జామినేషన్. కెన్ క్రాక్ ద నీట్ ఎగ్జామినేషన్ ఇన్ యువర్ ఫస్ట్ అటెంప్ట్. ఓకే, ట్రై టు సాల్వ్ ఆల్ ద క్వశ్చన్స్, MCQs. ఓకే, యూ కెన్ ఫాలో అవర్ ఛానల్. ఫస్ట్ ఇయర్ సంబంధించిన అన్ని చాప్టర్స్ని మనం కంప్లీట్ చేసి పెట్టేసాం ఆల్రెడీ. టోటల్ చాప్టర్స్ ఫ్రమ్ లివింగ్ వరల్డ్ టు కెమికల్ కంట్రోల్ అండ్ కోఆర్డినేషన్, ప్రతి చాప్టర్ నుంచి 30 క్వశ్చన్స్ సెలెక్ట్ చేసి ఎక్స్ప్లెనేషన్తో సహా మనం ఛానల్లో పెట్టాం. సెకండ్ ఇయర్లో కూడా రిప్రొడక్షన్, రిప్రొడక్టివ్ హెల్త్, ప్రిన్సిపల్స్ ఆఫ్ ఇన్హెరిటెన్స్, మాలిక్యులర్ బేస్ ఆఫ్ ఇన్హెరిటెన్స్, ఇవి కూడా అవుతున్నాయి. రేపు నెక్స్ట్ చాప్టర్ చూద్దాం. సో వన్ టర్న్ ఆఫ్ ద హెలిక్స్ ఇన్ ఎ బీ ఫార్మ్ DNA is approximately 27 says, okay, Evergreen says D, Bargo says C. 0.34 nanometers is between two adjacent nitrogen bases. It is 0.34. But total strength, total length, helix length, it is 3.4 nanometers. So Evergreen correct, perfect. You know, angstroms. How many angstroms length it is? It is 3.4 nanometers. Okay. If you want to convert this nanometers into the angstroms, how many angstroms length it is? It is 34 angstroms. అది నేను మీకు అనౌన్స్ చేస్తాను పూజిత. ఏ రోజుది ఆ రోజు నేను చెప్పగలుగుతున్నాను, కానీ ముందు రోజు ఇంతకు ముందులా చెప్పలేకపోతున్నాను. ఇంతకుముందు అంటే చెప్పేవాడిని ఎవ్రీడే ఈవినింగ్ 4, ఈవినింగ్ 6 అని. సో డిపెండింగ్ అపాన్ ద కాలేజ్ టైమింగ్స్ అండ్ మై అదర్ వర్క్స్ ఆల్సో. స్టూడెంట్స్కి నోట్స్ అది రాసేవాలనుకుంటున్నాను. సో వాళ్ళకి ఇచ్చే నోట్స్ రాయడానికి, ఇంకా కొన్ని పర్సనల్ వర్క్స్, ఇవన్నీ అనలైజ్ చేసుకుని ఒక టైం కుదిరిన తర్వాత అది 2 డేస్కి ఒకసారి, ఎవ్రీడేనా 3 డేస్కి ఒకసారి చూసి చెప్తాను. ఓకే, నో ప్రాబ్లం. ఇంకా ఫస్ట్ ఇయర్ కాబట్టి ఓకే, నో ప్రాబ్లం. సెకండ్ ఇయర్ వాళ్ళకి కొంచెం ఇబ్బంది పడవచ్చు. Telomerase is an enzyme which is a what? Telomerase is an enzyme which is Very good, Avery. That is 34 angstroms. So అప్పుడు నువ్వు చెప్పింది 3.4 angstroms between two adjacent nitrogen bases. 10 nitrogen bases are there. So 34 by 10, 3.4 angstroms between two adjacent nitrogen bases. That is one. And if we want to convert it into the nanometers, it is 0.34 nanometers. Total length 3.4 nanometers, like that. For 28th question, what is the right answer for 28th question? Telomerase is an enzyme which is enzyme. So it is a protein. It is not a simple protein. It is a ribonucleoprotein. Ribonucleoprotein it is. Ribo sugar. Okay. Next. That means what? The protein which have the capability to do the transcription, translation, replication, this type of activities in the DNA material, in the genetic material. The type of protein is ribonucleoprotein. Nucleoprotein it is, and that is the telomerase. 29th question. One gene, one enzyme hypothesis was postulated by one gene and one enzyme. One gene and one enzyme hypothesis is postulated by లావన్ అంటాం కదా మనం క్రికెట్లో. ఇంకొక క్వశ్చన్ ఉంది కంప్లీట్ చేసుకుంటే. 29th D. Garrod is wrong, Saujanya. Sorry. They are two scientists, Beadle and Tatum. Beadle and Tatum, they explain this. What it is, when they observe one type of chemical pathway. Chemical pathway means there is a series of reactions, one by one chemical reactions are there. In that one by one chemical reaction, series of reactions, some enzymes are participated. Star enzyme, circle enzyme, triangle enzyme, square enzyme. This type of enzymes are participated in that pathway in every stage. Okay, and every enzyme is coded by a particular gene. This is a gene, and this is another gene. This is another gene. This is another gene. So one gene, one enzyme. They observed that, and they give the hypothesis as one gene and one enzyme hypothesis. They are Beadle and Tatum. Okay, come to the last but not least important question, that is, which one of the following makes use of RNA template to synthesize DNA? Very interesting. Which one of the following makes use of RNA template to synthesize DNA? Normally DNA template is there. From the DNA template, RNA is prepared. This is called transcription. Now the question, which one of the following makes use of RNA template? It is RNA template, for example, to make DNA. That means what? Reverse to the transcription. Which enzyme is that? Is the DNA polymerase or RNA polymerase or reverse transcriptase or DNA dependent RNA polymerase? Yeah, everyone should answer this question. Very, very easy question. Interesting question. Okay, you know, it causes one deadly disease. No Bhargav. Just say that from DNA, RNA, this is transcription. Reverse to the transcription, that is reverse transcriptase. Reverse transcriptase. And it is present in HIV. Human immunodeficiency virus have an enzyme that is reverse transcriptase. Means what? Transcription process is in reverse manner from RNA to DNA. This is reverse transcriptase enzyme. Okay, and that will form the DNA from the RNA. And you know, this parasite is called smart parasite. Is smart Shankar, smart parasite. HIV. So this is about today's session, and I hope you enjoy, and thank you so much for your participation, and make the session successful. You make the session successful. Thanks for that. and try to follow me in different social media sites. Of course, I am not active in Twitter, and blogger is also not yet completed, but notes అది అప్లోడ్ చేశాను నేను బ్లాగర్లో. మీరు అబ్జర్వ్ చేయొచ్చు. రాజేష్ బయాలజీ బ్లాగ్ ఓపెన్ చేస్తే rajeshbiology.blogspot.com, అందులో కొన్ని నోట్సెస్ వరకు ఉంటాయి. బట్ టెలిగ్రామ్లో నేను లైవ్ సెషన్స్ కండక్ట్ చేసేటప్పుడు మీకు రెగ్యులర్ అప్డేట్స్ ఇస్తుంటాను. ఏదైనా నోట్స్ ఉంటే కనుక దాంట్లో పోస్ట్ చేస్తుంటాను. అండ్ ఇన్స్టాగ్రామ్, యూ ఆల్రెడీ నో వాట్ హ్యాపెన్ ఇన్ మై లైఫ్ అవుట్ ఆఫ్ ద స్క్రీన్, అవుట్ ఆఫ్ ద థింగ్. అండ్ నోట్స్ ఈజ్ ఆల్సో ఇఫ్ పాసిబుల్, ఏవైనా మంచిగా ఉంటే కనుక వాటిని నేను షేర్ చేస్తుంటాను. అండ్ కమ్ టు హియర్, దిస్ ఈస్ ద స్టూడెంట్ పల్స్. వీఆర్ గోయింగ్ టు బిల్డ్ దిస్ వెబ్సైట్. ఇది అండర్ ప్రాసెస్లో ఉంది. studentpulse.online త్వరలోనే లాంచ్ చేయబోతున్నాం. అండ్ దీంట్లో అన్ని రకాల కోర్సెస్ని ఇవ్వడానికి ట్రై చేస్తున్నాం. ఫిజిక్స్, కెమిస్ట్రీ, మ్యాథమెటిక్స్ అండ్ తెలుగు అండ్ డిఫరెంట్ లాంగ్వేజెస్. డిఫరెంట్ లాంగ్వేజెస్ మీన్స్ మీరు స్పానిష్, ఫ్రెంచ్ వీటిని ఇమాజిన్ చేయొద్దు. నా ఉద్దేశం ఇంగ్లీష్, సంస్క్రిట్, తెలుగు, ఇలా మనకి ఇంటర్మీడియట్లో ఉండే లాంగ్వేజెస్. ఫిజిక్స్, కెమిస్ట్రీ, బాటనీ, జువాలజీ, సివిక్స్, కామర్స్, ఇలా అందరు లెక్చరర్స్లో కోఆర్డినేట్ అవ్వడానికి ట్రై చేస్తున్నాం మేము. లాస్ట్ 1 year నుంచి చెప్తున్నాను ఈ విషయాన్ని. కాకపోతే ఇప్పుడు ఇంకొంచెం ప్రాసెస్ అనేది స్పీడప్ అయింది. ఓకే, అండ్ if you want any tuitions or anything, you can give the information to this email ID. బట్ సారీ, ఇది కూడా మీకు చెక్ చేయడానికి టైం లేకుండా పోతుంది మాకు. సో మీరు యూట్యూబ్ లైవ్ సెషన్స్కి అటెండ్ అయ్యి కావాల్సిన ట్యూషన్ మెన్షన్ చేస్తే టీచర్కి మీ ఫోన్ నెంబర్ ఇవ్వడం కానీ, మా సార్స్ మొబైల్ నెంబర్స్ మీకు ఇవ్వడం కానీ చేస్తాం. అది ఫ్రీ, ఇదంతా కూడా ఫ్రీ కన్సల్టెన్సీ. సో థ్యాంక్ యూ సో మచ్. ఎవర్గ్రీన్ సార్, జనరేషన్ కండక్షన్ ఆఫ్ నర్వ్ పల్సెస్. ఎవర్గ్రీన్ ఒకసారి మన యూట్యూబ్ వీడియోస్లో ప్లేలిస్ట్ ఓపెన్ చేసి నర్వస్ సిస్టమ్ చూడు, దాంట్లో ఖచ్చితంగా నేను ఇది ఎక్స్ప్లెయిన్ చేస్తాను. జనరేషన్ అండ్ కండక్షన్ ఆఫ్ నర్వ్ ఇంపల్సెస్ అనేది ఆల్రెడీ మనం కంప్లీట్ చేసుకున్నాం. టైప్స్ ఆఫ్ గేట్స్. ఏ గేట్స్? దట్ మీన్స్ ప్రోటీన్ ఛానల్స్, అయాన్ గేట్స్. ఇవి సోడియం యాక్టివేషన్ గేట్ అండ్ ఇన్యాక్టివేషన్ గేట్, పొటాషియం యాక్టివేషన్ గేట్, ఇవి నేను చెప్పాను. జనరేషన్ అండ్ కండక్షన్ ఆఫ్ నర్వ్ ఇంపల్సెస్ ఉంటుంది. ఓకే, ఇఫ్ యూ వాంట్ టు సీ నౌ, మీకు చూపిస్తాను నేను ఇప్పుడే. వీడియోస్లో ప్లేలిస్ట్లోకి వెళ్ళండి, రాజేష్ బయాలజీలో. అందులో మనకి నర్వస్ సిస్టమ్ చూడండి. హియర్ న్యూరల్ కంట్రోల్ అండ్ కోఆర్డినేషన్. న్యూరల్ కంట్రోల్ కోఆర్డినేషన్లో జనరేషన్ అండ్ కండక్షన్ ఆఫ్ నర్వ్ ఇంపల్సెస్ ఉంది. ద పెరిఫెరల్ నర్వస్ సిస్టమ్. ఓకే, న్యూరల్ కంట్రోల్ అండ్ కోఆర్డినేషన్, ద సెంట్రల్ నర్వస్ సిస్టమ్, న్యూరల్ కంట్రోల్ కోఆర్డినేషన్. యాక్షన్ పొటెన్షియల్ అని ఉంది కదా, యాక్షన్ పొటెన్షియల్ 1 hour క్లాస్ ఉంది. ఈ 1 hour క్లాస్లో యాక్షన్ పొటెన్షియల్కి సంబంధించిన క్లాసెస్ ఉంటాయి. మ్యాగ్నెటిక్. And then next, these are the channels. Leakage channels are there. These are the channels, leakage channels. So activation gates, potassium gate, potassium and sodium gates explain చేశాను ఇందులో. ligand gated channels ఏంటి, different types of channels. ఓకే, నర్వ్ ఇంపల్సెస్ కనిపించడం లేదా? ఇప్పుడు నేను మీకు స్క్రీన్ మీద చూపిస్తున్నాను కదా, అయినా కనిపించడం లేదా? రీపోలరైజేషన్. వాట్ ఈస్ హైపర్ పోలరైజేషన్? ప్లేలిస్ట్లో ఉంది. ఇది ప్లేలిస్ట్లోనే ఉంది ఎవరికి, ఇది బయట లేదు. న్యూరల్ కంట్రోల్, యాక్షన్ పొటెన్షియల్ అని ఉంటుంది. డోంట్ సెర్చ్ యాజ్ గేట్స్, డోంట్ సెర్చ్ యాజ్ జనరేషన్ అండ్ కండక్షన్ నర్వ్ ఇంపల్సెస్. యాక్షన్ పొటెన్షియల్ అని సెర్చ్ చేయండి, మీకు కనిపిస్తుంది. యాక్షన్ పొటెన్షియల్ విచ్ ఈజ్ అన్ యాక్షన్. ఓకే, విత్ డిఫరెంట్ కలర్స్తో కూడా ఎక్స్ప్లెయిన్ చేసినట్టే గుర్తు నాకు. ఇది స్క్రీన్ మీద కనిపిస్తుందేమో కదా, is it not visible? And our stage వచ్చింది కదా, then it is ready to activation. More sodium enter into the exoplasm like this. Okay, right. More sodium into the exoplasm. Thank you. And thank you so much. It prepare or it convert the inside voltage into. We will meet in next live session. Thank you so much. Have a nice day and good night.